plans for that for this year. Now, it gives me pleasure to introduce Paris Papagianis, who will be formally introduced by Jane Burke, who, as you know, is based in Melbourne and has been doing a lot of innovative work for quite some time now in a range of areas, airways and pulmonary fibrosis, and has now become more and more involved in the area of silicosis, which, as we all know, is a very topical industrial health and safety issue at the present time. And so we look forward to hearing from initially Jane, who will introduce Paris. Thanks very much, Jane.
Thanks, Paul. And it's really pleased that Paris has got this opportunity as a Create Fellow to present some of the work she did before she joined my lab, but also some ideas that we've got that we're working on in this Victorian cohort of engineered stone workers. So Paris did a joint PhD across...
transplant fellow.
Let's see if we can
mute everyone else.
Just mute everyone, yeah.
Thanks. So Paris only finished her PhD a few years ago. She did a joint PhD between Monash and with Jane Pillow in Perth, and was doing some work in early life lung diseases and had a very successful PhD. She then did a postdoc at RIT, which is some of the work she'll present today, with Steve Bozanovsky on an MRFF funded silicosis grant. And then I've been really lucky that she's been able to join us. She's been in my lab now for the past year. It's really energised our group in this space with a lot of initiatives that have led to successful grant applications, and particularly in this silicosis space. So we started working on silicosis a few years ago with a pilot grant from Maurice Blackburn through TSANZ. That was an occupational lung disease small grant, and recruited a PhD student, Claudia Sim, some of whose work will be presented today. And that work underpinned some recent successful grants that we've had, and that hopefully will continue to bring in funds. As Paul said, this is such an important area. So as I said, it's great having Paris as part of our team, and I'm sure she's going to go from success to success, not only in this space, but more broadly as an early career researcher. So over to you, Paris, to tell us all about what you've been up to. Thank you.
Thanks, Jane. Thanks for a lovely introduction. I just want to confirm that everyone can hear me before I start. Perfect. So today I'm going to be outlining some work that I've done across RMIT and now at Monash University, looking at a particular cohort of Victorian workers exposed to engineered stone who develop silicosis. I want to start with an acknowledgement to country. So I recognise that the work I conduct is on the unceded lands of the people of the Kulin Nations, and I pay my respects to their elders past, present and emerging, and I extend that to the entire Aboriginal communities in Australia. So silicosis is an occupational lung disease. It's caused by the inhalation of tiny silica particles, hence its name. And just to give you an idea of what makes up an inhalable particle, the largest inhalable particle is around 100 microns in diameter. And those that are up to 30 microns in diameter can enter the thoracic fraction of the lungs. And those that are less than 10 microns in diameter can enter the respiratory bronchioles in the lungs, down at the level of the alveoli where gas exchange occurs. And this is where silica particles or silica dust fit. So just as a point of reference, a single grain of table salt is about 100 microns in diameter. So these silica particles are very, very small. And when inhaled, they lodge very deep in the lungs, in the most delicate parts of the lungs that are responsible for gas exchange. There's been an increase in the rate of silicosis cases associated with engineered stone in the workplace. So this lovely artificial marble benchtop, which has been kind of branded as Caesar stone, is safe when installed, but it's actually the cutting, grinding, and manipulation of these stones on work sites that creates tiny silica dust particles. And that's because these stones, artificial and engineered stones, are made up of up to 98% silica. So when these dust particles are inhaled in the workplace, like I said, they lodge very deep within the lungs, and they sit there and they cause scarring of the lung tissue. There's absolutely no cure for silicosis unless you consider a lung transplant at end-stage disease a treatment option. Silicosis is diagnosed by X-ray or CT scan. So here we have an X-ray of a healthy lung. The air is shown in black here. So this individual has air-filled lungs, as we would expect. And then in the advanced silicosis X-ray seen here, we see all these white speckles or nodules in the lung, which indicate massive fibrosis in the lung, which impedes gas exchange. And this is due to the inhalation of those silica particles. At a cellular level, we think that once inhaled, these silica particles are phagocytosed by alveolar macrophages and in an attempt to get rid of these silica particles, but they actually can't be broken down. So the alveolar macrophage kind of loads up with silica particles and eventually apoptoses and dies and releases all those silica particles back into the alveolar space. And so you get this continued cycle of the silica particles being passed around alveolar macrophages that causes inflammation in the lungs and eventually fibrosis. So this idea of silicosis is not a new one. In 1871, silicosis was named as a lung disease, and in the 1930s, it was recognized by the International Labour Organization, who, as an organization, tried to implement the requirement to wear PPE in the workplace. So here is an image of workers just before World War I, actually, who have respiratory masks on at the workplace to try and prevent the inhalation of silica dust. The International Agency for Research on Cancer reported the carcinogenic effects of silica in 1987, and in 1995, the World Health Organization announced an elimination of silicosis initiative to try and incorporate better work-safe practices. I just want to reiterate here that all of this is occurring before the introduction of engineered stone into the workplace. So we're seeing silicosis as early as the 1900s occurring from other occupations like mining or quarry work. And it's not until 2012 that we see the first case of engineered stone-induced silicosis in Israel. The first case of engineered stone-induced silicosis in Australia is in 2015. And in 2017, this is engineered stone-induced silicosis is recognised as by the Global Burden of Disease. In February of this year, the federal government, along with the Lung Foundation and co-chairs Dr. Ryan Hoy and Kate Cole, released the National Silicosis Prevention Strategy. And one of the key themes in this strategy and action plan is to try and eliminate new cases of silicosis from engineered stone emerging by 2028. And we collaborate frequently with Ryan and Kate. So the global rates of silicosis, this is a report released by the Global Burden of Disease in 2017, show in purple those countries that have not reported silicosis cases or have very few silicosis cases. And then in the red and orange and yellow, we see high rates of silicosis in these countries. And these are mostly in developing countries where it's unlikely that workers are strictly complying to PPE or have work-safe measures in place or air monitoring in place across the country. Alarmingly, we see that in Australia, we have quite an unacceptable amount of silicosis cases, considering that we are a developed country. So, as I've mentioned, in Australia, we see the first case of engineered-induced silicosis in 2015. But by 2020, there's over 600 cases across Australia. Now, this translates to 1 in 4 workers who use engineered stone being diagnosed with silicosis in Victoria, and 1 in 5 workers who use engineered stone being diagnosed with silicosis in Queensland. So really unacceptable rates of silicosis emerging from those workplaces that use engineered stone. Most of these individuals with silicosis are male. They're relatively young and they're considered otherwise healthy. So they very rarely have other comorbidities. By definition, silicosis can be categorized into 3 groups. The first is acute silicosis that occurs after a very short period of exposure to very high concentrations of silica dust. The second is called accelerated silicosis, which occurs over a slightly longer period of time, but with still very high concentrations of dust which accumulate over that time period. And then we have chronic silicosis, which is thought to occur over a longer time of exposure, but at more moderate levels. And just to complicate this further, we see that silicosis can be classified as either simple or complicated, and this is done by the use of X-ray or CT scans. So we're now looking at CT scans of the lungs, and in this example of an individual with simple silicosis, we see these small white specks here that are nodules in the lung less than 1 centimetre in diameter. In complicated silicosis, we get this progressive massive fibrosis in the lungs. So we see these huge nodules in the lungs that correlate with fibrotic lung disease, and there's a high risk of developing cancer in these patients, lung cancer in these patients. What is currently thought is that this simple silicosis represents a kind of early stage of disease where nodules are small, and then the individual can progress to a more complicated form of disease with these larger nodules, and this is normally accompanied by respiratory failure or a decline in lung function. So what I've shown you here is how to diagnose and detect silicosis disease that is already established. So hopefully what I'll touch on today is both how this disease occurs in the lungs, whether that be simple or complicated silicosis, and then ways that we could potentially detect this disease before we're seeing nodules on X-ray or CT. So screening processes in Victoria for those that work with engineered stone are facilitated by WorkSafe Victoria, who identify those at risk of developing silicosis due to their occupational setting and workplace air monitoring. Excuse me. So if you are identified as an individual at risk of developing silicosis due to exposure to engineered dust from engineered stone, you'd be referred to get CT or X-rays and lung function tests. And the cohort that we're looking at get this done at the Alfred Hospital. If your diagnosis is quite clear, then you get ongoing clinical care. But if you have an unclear diagnosis of silicosis, you can be sent to get further testing done. And in this case, this was a group sent to, this cohort is sent to the Austin Hospital. So they get endoscopy done so they can collect fluid from the lungs, so called bronchoalveolar lavage, so lung fluid samples from the lungs, and biopsies of both lung tissue and lymph tissue, as well as some bloods. So these samples are collected by researchers at RMIT and at Monash University. And as mentioned, my previous appointment was at RMIT, where I started working with this cohort of silicosis patients. And I'm now at Monash University continuing to work with this same population of patients. So at RMIT, we began to classify these patients with silicosis from engineered stone. And this is work that I really did in strong partnership with Dr. Christian Allo, who I think is online today. And so here we just have the demographics of those patients, and this is actually published now. So we have those with simple silicosis, so small nodules on CT scans, and those with complicated silicosis, so progressive massive fibrosis on CT scans, and then those with no disease. Now, because these individuals are also getting a bronchoscopy and a biopsy, no diseases are not strictly healthy controls. They are individuals who are undergoing a bronchoscopy or biopsy for another reason. So one of those reasons may be, for example, chronic cough. So we can see that all of those individuals with silicosis are males, and most of those without silicosis disease are male, and there's 2 females in that group. The median age in those that have simple silicosis is around 40 years old, and those with complicated silicosis is 44 years old. But we have an individual here as young as 27 that has progressive massive fibrosis in the lungs, which is quite alarming. We see also that most of those individuals with silicosis are current or ex-smokers, but the average amount of time that we expect each of these individuals with silicosis to have been exposed to silica from engineered stone is 13 years. And just challenging these traditional classifications of silicosis. We have someone here with progressive massive pulmonary fibrosis that was only working with engineered stone for 4 years. So despite the classification of complicated silicosis being a longer time to present, we're not seeing that necessarily with engineered stone. So then what does the lung function in these individuals look like? We have those no disease individuals in black here, the simple silicosis individuals in blue, and the complicated silicosis diagnoses in red. I've got the predicted DCLO here, which is an indication of the efficiency of gas exchange between blood and air. So looking at more of the respiratory bronchioles in this lung function test. And this dotted line here is the lower limit of what would be a normal range, which sits at about 75%. So anything above 75% is considered normal predicted DCLO in this case. So those individuals that have simple silicosis or complicated silicosis have lower predicted DCLO compared to non-disease controls, but we don't see a difference between this simple and complicated silicosis in terms of lung function. What's surprising here is that actually about half or a bit more than half of these individuals with silicosis actually have what would be considered normal lung function. So even though they have nodules on X-ray and are diagnosed as having silicosis, they're not necessarily showing any respiratory like lung function decline, sorry. We've also got here the FEV1 to FVC ratio in these individuals. Again, the dotted line is the lower limit of normal, and we only really see one patient that has poor FEV1 to FVC ratios in the simple silicosis group. There's no difference across groups. And what this ratio is telling us is the amount of obstructive or restrictive disease in the upper airway. So they don't appear to have obstructive disease. We were able to opportunistically look at the bloods that were taken at the time these individuals were referred to the Austin. Again, I'm going to continue to use this color scheme and this dashed line as a reference for the upper limit of normal in these following graphs. What we can see is that ferritin has increased expression in the bloods of those with simple silicosis, but there's no difference between simple silicosis and complicated silicosis. We see also an increase in the amount of iron in the blood, both in simple and complicated silicosis. And this became of interest to us because in other fibrotic diseases of the lung, and even in long COVID, we see an increase in iron in the systemic circulation of these individuals. And we also know that ferritin can be associated with decreasing DCLO predictions on lung function tests in those with pulmonary fibrosis from other origins. So if you do a PubMed search and you look at pulmonary fibrosis and DCLO, you get over 6,500 PubMed results. If you then couple this with iron or ferritin, you get more than 2,000 results and more than 700 results when you look at ferritin in association with pulmonary fibrosis. So it's clear that iron and ferritin have an important role in the lungs. Iron is an essential nutrient for normal cellular processes in the lungs. It's sequestered by lung macrophages, and through ferritin within the lung macrophage, ferritin is then able to store and release iron as needed under normal homeostatic conditions. This is done through the release of oxygen species. Now, logic will then tell us that if you have more iron in the circulation, there's more reactive oxygen species in the lungs, and this can spill out of those macrophages and interact with the surrounding tissue to potentially cause fibrosis in the lungs. So we wanted to try and understand where the elevated iron is coming from. And so we looked at the total iron binding capacity in these individuals, and this was upregulated in both the circulation of the simple silicosis individuals and complicated silicosis individuals. But again, we don't see differences between simple and complicated silicosis. We still at this point are not clear on whether or not there is iron overload despite there being more iron in the systemic circulation. So we looked at the transferritin saturation, which is basically a ratio of the iron and the total iron binding capacity. And these two dashed lines represent the normal range in healthy individuals. And what we can see is that despite having increased ferritin and increased iron, these individuals with silicosis are not showing indications of iron overload. So where is this increased ferritin or increased iron coming from? We looked at inflammatory markers in the systemic circulation. So C-reactive protein, which is kind of a very standard broad inflammatory marker used in routine bloods, and also serum amyloid A, which is an indication of liver function in the systemic circulation. What we see is that these are not different between any of our groups, which indicates to us that the increases in ferritin and iron are probably not from the systemic circulation. They're probably not from liver dysfunction, for example. But it also tells us that markers within the blood, whether that be CRP or SAA or even ferritin or iron, are maybe not going to be able to predict those patients that have silicosis unless there's other comorbidities present responsible for those elevations in systemic markers. So we crudely looked at the amount of ferritin in the blood and the amount of iron in the blood, and they seem to correlate. So this confirms that there's a relationship between those two. And what we do know is that silica dust actually has a really high affinity to iron. So in these studies, they loaded silica dust with iron and then placed those iron-loaded dust particles onto human lung macrophages and then looked at the amount of inflammation or ROS being produced by these lung macrophages. And we can see that they're increased when you have this coupling of iron and silica dust particles. If you wet the dust, which is meant to mimic the safety procedures of the workplace, so workers are advised to wet the engineered stone as they cut it, we can see that there's less inflammatory markers being released by these lung macrophages and less reactive oxygen species. But alarmingly, there's still actually quite a bit of inflammation and ROS in these lung macrophages. So, this tells us that perhaps we don't have a very safe way of working with engineered stone yet. So if iron and silica have a high affinity and bind to each other, the transportation of that iron particle with the silica particle to the alveolar macrophage may be one that is supposed to be protective in the body. So once transported to the macrophage, this complex then interacts with ferritin, and ferritin produces excessive amount of reactive oxygen species and inflammatory markers in the lung as this silica particle can't actually be broken down. So we then became very interested in what the alveolar macrophages in the lungs of these patients looked like. So this is now from lung fluid washouts from these individuals. Now we noticed that through differential cell counts that macrophages were the dominant cell type in the lungs of these individuals. So there wasn't really huge differences in neutrophils, for example, in the lungs of these silicosis patients compared to non-diseased. What we do see in the non-disease group is relatively normal looking macrophages, and that in simple or complicated silicosis, you kind of get these really like vacuolated enlarged pockets in in these alveolar macrophages. And when we stain these with oil red ochre, we see that they're actually, in fact, lipid bodies within the alveolar macrophages. Lipid bodies within alveolar macrophages have also been implicated in idiopathic pulmonary fibrosis, so fibrotic disease of unknown origin in the lungs. And we think there's been reported also a relationship between excessive ROS and lipid production in alveolar macrophages. So if we then look at how much ferritin is in our lung fluid as opposed to the circulation of these silicosis patients, we see that there seems to be a correlation between the two. So Christian really nicely looked at the level of mRNA of ferritin heavy chain and ferritin light chain, both in the fluid and in the lung tissue biopsies of these individuals with silicosis, and saw that ferritin had higher expression in the fluid than in the biopsy. And what this indicates to us is that perhaps the lung itself is the source of this ferritin. So when iron and silica bind and are transported to the macrophage, ferritin works to try and sequester the iron and the macrophage tries to get rid of this silica particle. We get excess ferritin and ROS production, which spills into the vasculature. So we think that the rises in ferritin in the circulation are secondary to that of rising ferritin in the lungs itself. And so now I'm just going to move on to some really great work that's been done by Claudia Sim, a PhD student in our lab at Monash. She's been working on the same lung fluid bronchoalveolar lavage samples from this cohort and really trying to detail the silica that are present within individual macrophages in the alveolar space. So importantly, Claudia is able to confirm that these macrophages are filled with lipids, and they definitely don't look normal by any sense of the word. And then with polarizing microscopy, she's able to show where those silica particles lie in alveolar macrophages and actually just how small they are. So there's a particle here that's 0.24 microns in diameter. So really tiny, tiny particles lodged deep in the lungs of these silicosis patients. Claudia's then been using something called pseudo-color orientation to look at angles of diffraction when light is shone through these macrophages and passes through these silica particles. This tells us something about the density of the particle, and it creates like a color, a speckle here, and each different color represents a different silica particle. So Claudia is also a CREATE fellow, and I believe she's giving a talk in a few meetings time. So she'll be able to expand on this, and anyone that's interested in this, I really urge you to join that meeting. So Claudia then went on to try and look at the silica load within bowel samples from these patients. We see here that in the control macrophages, we don't see any silica particles using polarized light microscopy, but in those with silicosis, you can see those silica particles in the macrophage. Claudia calculated through a lot of counting the proportion of silica containing macrophages in the lung fluid samples of these patients, and alarmingly more than 50% of the macrophages present in these samples have at least a silica particle in them. Claudia also worked at looking at the silica load within the bowel of these people with silicosis. So looking at how many macrophages there are within the lung fluid sample, and then quantitating the amount of silica containing macrophages and particles per cell. And what she found was that there's actually quite a range of the average number of silica particles within these bowel samples, where some have macrophages that are loaded with silica particles, but most sit around that kind of 1 to 10 silica particles per cell. So to better understand what this means at an individual level, Claudia then looked at the number of cells per particle. So each of these dots represents a cell, the number of particles within a cell, and each of these are individuals from our cohort. And what you can see is that most alveolar macrophages have, 1 to 10 or 1 to 5 silica particles within the macrophages. But then you have these individuals with macrophages within the sample that are absolutely loaded with silica particles. If we go back then to look at what the radiological CT scoring told us about these individuals, we have all of our individuals diagnosed with simple silicosis here, so small nodules on CT, and those with complicated silicosis, so progressive massive fibrosis on CT, and then those that were determined to be exposed but didn't have any nodules on CT. What we see is that the range of silica particles per cell is much greater in those with simple silicosis compared to those that have complicated established progressive massive fibrosis in the lungs. We're not exactly sure what this means. It could be that if simple silicosis is an earlier stage of disease, there's more of an inflammatory process going on. The alveolar macrophages are more active than in complicated disease where there's more established fibrosis and lung function decline in these individuals. What is alarming, though, are these two individuals that were exposed to silica dust in the workplace, but had no lung function decline and no nodules on CT scan. They still have macrophages with silica in them. So this is a group that we're really interested in studying further. And so that brings me back to this idea of how the current screening system works in Australia. The workplace exposure surveys include things like how long the individual's been in the industry and what kind of PPE and how compliant they are in wearing that PPE. Again, the spirometry, which I've shown you, doesn't always detect a decline in lung function in silicosis patients, and also X-rays or HRCTs, which are used to identify established disease and not early disease. So in those individuals on the previous slide that were exposed to silica dust, but didn't have nodules, they would be determined as having no disease, even though they're likely at high risk of developing silicosis with ongoing work and ongoing exposure. On top of this, this screening protocol can be inconvenient and time consuming, so it requires hospital visits for spirometry and X-rays or lung function tests. And the HRCT scans are really great, but they also expose the individual to radiation, so you can't have more than 1 or 2 a year. So what we're now working towards is ways of non-invasively identifying those workers at risk. So we don't want to have to take bronchoalveolar lavage samples, for example, every time we need to confirm or identify a case of silicosis or those at risk of silicosis. This should be easy to apply, time-saving, and provide on-site screening. And importantly, it should be of low cost, because at the moment the cost lies with largely lies with the employer. And also the time away from work is a concern. So we're now beginning to do this at Monash University under Jane Burke, looking at chemicals in exhaled breath from this same cohort of people who work with engineered stone. And we're looking at something called volatile organic compounds in the breath. Now, VOCs can be influenced by exogenous sources, so how fit you are, your diet, or whether you smoke, any prescription medication that you're on, or your microbiome. And VOCs can also be influenced by endogenous sources, so your genetic makeup. Importantly, you can detect thousands of VOCs or chemicals in exhaled breath, and this is a non-invasive and quick analysis. So it's not a new idea, this idea of screening for disease in exhaled breath. It's been used a lot in gastrointestinal complications, and in particular in intolerances to food, in liver dysfunction and disease, and more recently, experimentally, at least in lung cancer and lung fibrosis. So the way that it works is that the individual breathes into a device which captures the exhaled breaths and the VOCs in the exhaled breath. Within a minute, you get an idea of one complete blood circulation around the body, and this gives us an idea of the VOCs transferred from blood to breath. If you can continue collecting samples in individuals for up to 10 minutes, you can detect those VOCs, which are normally have very low levels of expression. And these could be important in terms of biomarkers for identifying those at risk of developing silicosis, or for screening those people with silicosis in the workplace. So the technology already exists. There's a device called a breath biopsy, which is owned by Alstone Medical in the UK. And this is an image of it here. We have the mouthpiece, which the individual breathes into, these collection tubes, which collect the VOCs, and an air valve here, an air inlet here. So the way that this works in a bit more detail is this red arrow indicates breath from the patient. So this is exhaled breath. It's able to pass through this saliva guard, so we're able to remove any moisture, and then it passes through this system and out the air valve at the back. All the while air comes in through this inlet, and the individual's able to breathe consistently for, say, 10 minutes. The VOCs are actively pumped into these desorption tubes, and there's an electronic board here which can actually be manipulated to identify specific volatile organic compounds immediately, if you know what your targets are. So we are partnering with Alstone Medical to begin this breath testing in the already recruited individuals from this cohort with simple and complicated silicosis, but also extending this to those that are exposed to silica dust in the workplace, but don't have obvious signs of lung function decline or disease on X-ray or CT. Importantly, we will also be able to better match controls, because this is a non-invasive portable device. We're able to go to other work sites and age match and lifestyle match these individuals to healthy controls. So we would be collecting the samples and then analysing using relatively standard gas chromatography mass spec analysis, where we unbiasedly collate all the VOCs that we can detect in these individuals, and then look at which of those VOCs are differentially expressed between each of these groups, and then reconfirm by collecting additional breath samples that these differential expressions are true. Down the line, we can actually fit this breath biopsy device with specific sensors at the electronic board here highlighted in yellow, specific VOC sensors that we know to be associated with either of these groups. So hopefully, I should say that this work is all funded by the Victorian Medical Research Acceleration Fund, and we're hoping to begin this in June this year. So hopefully I've shown you that the current screening programs are only able to pick up established lung disease, and that lung function and no nodules on CT doesn't necessarily mean that you're at no risk of developing silicosis from engineered stone. What I'm now going to, and I've also shown you that the technology exists through Alstone Medical, and that we already have started recruiting some of these individuals. What I'm now going to show you is that you can actually detect VOCs in individuals with silicosis. So this is a study that was conducted in Iran in 2016. So this is just before the introduction of or the identification of engineered stone-induced silicosis. So these individuals have silicosis from other occupations. They also, in this study, looked at smokers that they deemed healthy or non-healthy. So non-healthy smokers had lung cancer, for example, and then they had healthy controls. And they came up with signatures of volatile organic compounds that they associated with different metabolic processes. So this first signature that they identified was to do with smoking, unsurprisingly. But the second signature was related to oxidative stress in the breath of individuals that had silicosis. So again, reiterating our findings in the alveolar macrophages, and also they saw lipid peroxidation markers in those with silicosis in those 4 samples as well. So again, reiterating our findings of these kind of lipid-filled alveolar macrophages in the lung that are dysfunctional and can contribute to inflammation and fibrosis. So in the future, workplace questionnaires to indicate the level of exposure are still going to be crucial, but perhaps we could couple this with breath sampling to identify those that are at risk of going on to develop silicosis. This would be much more convenient for individuals, as they don't have to visit hospitals for X-ray or lung function tests. If they are identified as being at risk of developing silicosis, it's at that point that they would then go to the clinic. And this would hopefully also decrease the long wait lists we see in clinics for screening people for silicosis. So we had our first case of silicosis in Australia from engineered stone in 2015, and we're now at this kind of unacceptable level or rate of diagnosis in workers that cut, grind, and drill into engineered stone. And what we're offering in line with the National Silicosis Prevention Strategy is detecting individuals at risk of developing progressive massive fibrosis in the lungs earlier, so that they can better manage their symptoms and their disease with clinical support over a longer period of time. And we would increase monitoring in those people that are at risk, but perhaps have been exposed to silica dust, but don't necessarily have nodules yet. And in this way, we're allowing for this cohort to potentially get to an older age and die with disease rather than die of the disease. And then we would be increasing on-site standard monitoring so that we can also reduce wait time for at hospitals, so that we don't have these huge wait times for X-ray and lung function. And I just want to finish off by saying that this work that we're about to start has actually been published in the Monash BDI Institute News today, and outlines the funding and what we're going to do. So if anyone's interested in that, you can find that on the Monash BDI website. And I just want to thank you all then and wrap up. I want to thank Christian especially, and Claudia, who did a lot of the work that I presented today, Jane for her supervision, all of the clinical partners, all the participants that offered samples for this study, and then all of the funding bodies as well. Thanks.
Well, thanks very much, Paris. We now have some time for questions and discussion. I might just sort of start the ball rolling and maybe be devil's advocate a little bit. I mean, there's no doubt that there's interesting science here, and that's great. But in terms of managing the condition, given that we don't believe there's any safe level of exposure to silica, we don't have any treatment for it when it happens, I don't know that we really know the risk of progression once you have, let's say, just a few macrophages with silica particles on your BAL and not much else. What are you going to actually tell the patient? So pragmatically, wouldn't we be better off just saving all that money and just banning silica stones?
The short answer is yes, we would be better off to ban engineered stone in the workplace. And that is something that's being pushed by the LFA, by TSANZ, and also Jane's represented the TSANZ on this, as well as Kate Cole and Ryan Hoy. So that in an ideal world, that would be great. We're still going to see cases of silicosis in other industries like mining, quarrying, tunneling, and also in those underdeveloped countries. So the use of a portable device actually is not restricted to those individuals that are working with engineered stone, we're using this cohort because they're so well defined. They've got lung function tests, they've got CT scans, they've got a really thorough history of their workplace exposure. And so we're able to get a lot of information from these people as a pilot in the initial period. And then the hope is eventually to expand that kind of breath test analyses to other industries and occupations as well.
Okay. Could I perhaps just get you to stop sharing your screen, and then we can see everybody's face on the computer, and it just makes me a little bit easier to spot who's got their hand up. So can I invite people to perhaps use the raise hand function so that we can keep a little bit of order going here? I might just pose another question, and this might be a little bit tangential, but I just coincidentally happened to be at a function sponsored by Turner Freeman last night, who, as you know, have had decades of asbestos-related litigation representation and now getting into silicosis. And they were telling me that the laws around silicosis are way behind at the moment, and you only kind of get one bite at the cherry to get some sort of compensation. And so they're agonizing about, you know, when people should put their hand up, because if they put their hand up and they've got mild disease and then it gets worse, they're kind of a bit snookered. Is your group, or, you know, advocating or getting involved in these discussions about changing the legislation about that?
Yeah, so we have just started beginning those discussions with people like the Lung Foundation Australia. Jane also has funding from iCare, which is an insurer in New South Wales, to discuss, you know, discuss these kinds of issues. We're not interested so much in the litigation side as opposed to preventing the amount of people that are going to need to go and seek litigation. But it's definitely an important issue. And just recently, Boral had to pay out an individual who worked in a quarry for the dust disease induced lung injury that they had. So I think watch this space, but it's not of particular interest to our research group, but something that we're going to pursue.
Fair enough. Now, so Joanne Dickinson has a hand up. Go ahead.
Thank you, Paris. That was really, really interesting. I'm a little bit interested in the mechanisms, and particularly the people that develop quite severe disease after a relatively short exposure. Versus, and I don't even know, so this is sort of highlighting my ignorance, but I don't even know, are there people in the industry who've worked with engineered stone or indeed exposure to silica for 20, 30 years and don't develop disease? And so what you think the mechanisms are around that. And then I also noticed that you've got a genomics arm to some of the research you're doing, and I wondered if you could expand a little on that too.
Yeah, sure. So the short answer is we don't understand how this progresses to the point of massive pulmonary fibrosis yet. There certainly are people that are exposed to very high levels of silica dust from engineered stone that are getting very complicated disease within a short time period. But there are also people that can work in this industry for many, many years and don't really have any symptoms, despite probably having inhaled silica dust. So they have quite a stable disease. So we don't understand how those two people progress. And that is something that we're really trying to work hard on. That's something that Claudia Sim, the PhD in our lab, is really working hard to try and delineate as well. But the short answer is, yeah, we don't know. There are some people that are stable and there are some people that really deteriorate very quickly. And normally those people that deteriorate quickly are smokers as well, or ex-smokers. But apart from that, we don't really understand why. We also have data on the estimated level of exposure, which I'm currently looking at now to try and see if the severity of the disease is associated with how long these people have been exposed to the dust or worked in the industry for. So hopefully we'll get a clearer answer with that data. And I think, yeah, I was going to say something else that I've forgotten now, so that's fine. And then your final point about the genomics arm. Yeah, so that was more a comment that your genetic makeup can influence what volatile organic compounds are expressed in your breath. And we won't really understand that until we're able to collect the samples. I know that Steve Bosanowski and Christian's lab are looking at RNA-seq in some of these individuals. So that will give us a much better idea of how complicated this silicosis disease is across a simple and complicated diagnoses.
That's really interesting, and I think that maybe that there and this is probably because of my background, which Paul knows, is that I'm quite interested in the genetic predisposition to pulmonary fibrosis. And, and I wondered, there's a little bit in the literature about some of the genetic variants that are associated with development of fibrotic lung disease, such as, you know, the MUC5B, which is probably the strongest common risk variant. And whether or not you think, you know, because that's certainly something that influences alongside smoking, you know, the development of fibrotic lung disease in other types of interstitial lung disease. And so I've thought about, you know, doing that sort of work too.
Yeah, so there's a study out of China that looked at chromosome variation in individuals with silicosis from mining, and they identified an abnormality, I can't remember the details, but an abnormality on a chromosome that they associated with severe disease in that cohort. I know that when I was at RMIT, Christian looked into whether or not there was an association between disease severity and potentially that alteration in the chromosome. We didn't see anything, at least in engineered stone-induced silicosis. So whether or not there's slightly different pathophysiology between diseases depending on what the source of silica is one possibility as well. So this becomes quite, it becomes quite complicated. You know, how freshly fractured is the particle that you're inhaling? How big is it? Where's it coming from? Is it engineered stone? Is it mining? Is it sand? I mean, and these things will probably reveal slightly different types of diseases, or at least breath markers, which is our main interest.
No one else has got their hand up at the moment. I
think Adrian.
Oh, Adrian, is that a hand?
Yes, hi. So I'm Adrian Edwards. I'm a respiratory physician in Christchurch. So I was just wondering, with your exhale breath, is there a way of actually just looking at exhaled breath macrophages? Has that been done before? I know you can do it with the eosinophils. So just in, you know, sampling sputum, or an easier way rather than doing a BAL.
Yeah, sure. So we have a collaborator in Israel called Lizzie Fireman, who looks at, she was the first one to identify engineered stone-induced silicosis in Israel, and she looks at sputum from patients with silicosis. It's not a very nice procedure. It's, you sit there and you kind of hack up all the saliva and spit it in a tube. So we will, and we plan to compare our breath with sputum because it's also non-invasive, to try and understand if one is more predictive of risk of disease than the other. But the simplicity of just using a breath test and capturing tiny particles in the exhaled breath for on-site screening in the first instance is really attractive to us. And we've had really good feedback from other respiratory physicians in Victoria in particular, and in New South Wales. So I think that's one point. The other thing is that these VOCs are really, really tiny. So with this device, we wouldn't be able to capture eosinophils or macrophages or any of those things. So we actually are looking at, we're not capturing cells, we're capturing tiny chemicals. We're capturing something smaller than droplets in condensate. So yeah, with this device, we won't be able to do that.
Given the impact of the particles on the macrophages, and obviously macrophages are an important defense mechanism, I mean, intuitively you might think people would be at a greater risk of chest infections, but I don't know if there's data on that, and whether there's any third world data about the link between this and tuberculosis, because I think there was data back in the old days with the coal miners and TB. What's the story there? Yeah,
sure. So a really good point. We in this cohort have excluded for those that have secondary infection like tuberculosis, but we do know that these individuals that have silicosis are at much higher risk of respiratory infections and pneumonias, especially repeat respiratory infection, and pneumonias. There are studies that look at markers in blood and in breath in silicosis from other occupations, and they have identified what they think are markers in breath and blood, but these individuals in the study often also have tuberculosis or something else. So it's not clear whether or not the markers are from the infection or from the underlying disease. So in the first instance, I think it'll be important to separate those two, but definitely these patients have increased susceptibility to respiratory infections.
Yeah, okay. Any other questions? I think I raised this question at the last meeting. It might have been about whole lung lavage as a treatment. What are your group's thoughts on that one? I think there's a bit going on in Melbourne, isn't there?
Yeah, so there's an MRFF held by Steve Bozanowski and Dan Chambers looking at whole lung lavage in these in silicosis patients. It's largely for people with established disease, so they've been diagnosed with silicosis and they have the early stages of the disease, so they're trying to wash out any silica particles in the lung. What we're looking at is I think something that's going to identify a risk factor for an individual. It's quite a laborious procedure as well, and I think one of the points risen with this breath test is the convenience and the time-saving capacity of it as well. I do know that they do quite a lot of whole lung lavage up in Queensland, especially with Dan Chambers.
Yeah. All right. Well, we're coming close to time. Can't see anyone's hand up. Jane, did you want to have any final words?
Just to comment on the TB interaction and stuff. So there's now quite a lot of aid agencies are setting up screening for silicosis and TB in third world countries as simultaneous screening programs. So in terms of, whether this might end up being something with broader application beyond engineered stone, Victoria, Australia, it's sort of quite exciting to think about something that could be introduced more widely as a part of a aid type screening in countries where there are minors attending clinics for the TB and having potentially this done at the same time. So I think it's really exciting. It is very early stages, but we think we've got a lot of the stakeholders engaged in this and quite enthusiastic to take it through. And we've also got, support from people with expertise in exhale breath, because this is a little bit outside our normal scope of things. We're still developing our lung slice models, extending that to silicosis as well, to be looking at initiation of disease at that sort of cellular level and potential interventions for those changes. So it's great to have a good team, a little bit of funding, never enough, but we're heading in the right direction with this work, we think. And it's great having Paris and Claudia and another colleague, Simon, as part of our team. So thanks, everybody.
Yeah, no, great. Good. Very important.
Paul, I'll just jump in and say there is one more question from Alexandra Muthu.
There we are. Alexandra, I saw you there. Go ahead.
Hi, sorry, I obviously had my video off, so you didn't see me. My question is, Paris, if you wouldn't mind just defining in your initial set of data, you had the no diseases, and I heard you say that they were people that were coming for respiratory endoscopy for unrelated causes, but I wasn't sure if they were also confirmed as having no industry exposure to silica dust. If you could just clarify that, thanks.
Yeah, sure. So they are confirmed as having no exposure to silica dust. So they're not from the workforce at all. They're people that are presenting for a biopsy or endoscopy for an unrelated matter. So chronic cough was a common one. And then I can't remember the other ones now. We've only got about 5 or so of them. They're very hard to recruit. But yeah, they're totally no known exposure to silica dust.
Okay, so was there a reason why they weren't called controls?
Yeah, because I think that they're not strictly healthy controls. Ideally, we would want a sample of healthy bowel or a healthy biopsy to compare to. There's probably a lot of underlying inflammation in those individuals that are not silicosis disease. So I think for that reason, you can't call them controls strictly.
Yeah. What's going to be good, though, for the breath sampling is that because it's non-invasive, we should be able to get true controls for those studies, which will be really, really increase our power of working out differences. So,
All right. Well, that pretty much nicely takes us right up to time. So thanks again to Paris and Jane for that interesting presentation. You're certainly on the crest of a wave there, and great to hear all the grant success and things coming out of it. So we look forward to seeing a lot more coming. So thanks very much, and we'll see you all for the next meeting. We'll get the notices out in due course, and we might even see some of you at the ATS. I'm not sure. Okay, bye now.
Thank you.